So I apologize to you guys. I thought I'd sent the slides yesterday morning, and Dr. Pearson rescued me as usual. He's a fantastic
block here.
He's also, I have to be careful because he's my collaborator as well, so sorry about that. You're right. But anyway, so today I'm actually going to talk to you about something that is kind of my passion for what I do clinically. I'm going to talk to you about infections of the brain. You know, you hear a lot about non-communicable diseases like stroke and epilepsy. And so today, we're going to talk about communicable diseases. And that's pretty important because we are in an era today of emerging infections. Every year for the last decade in Canada, we've had a new infection emerge. Whether it's listeria, mad cow disease, H1N1, SARS, you name it, we've had it in Canada. So it's an area that actually is important because it can have devastating effects in a very short period of time. The best example is SARS. So some of you will know the story of SARS. It's a virus. It was first recognized, or it probably originates in agricultural markets in Southeast Asia, probably in southern China. It was first recognized in Hong Kong. It was Hong Kong. And there were several patients who were sick in a hotel. And within literally a day, there were people sick in Toronto. Someone had flown from Hong Kong to Toronto and spread it in a hotel elevator. And then it became, as you probably know, people died from it, and it was turning the city of Toronto upside down. And it really had immense social, economic, and of course medical implications. So that's the kind of time period we're dealing with infections today. And it's very different from 100 years ago or even 20 years ago, where it took us a long time to travel around the world. There are a lot of other things that have contributed to infections. Climate change is a reality. You hear about Ebola virus infection in Africa, and that's in part due to the forestry industry. You also hear about infections like HIV, which I'm going to talk to you about today, where it's as consequences of changes in social, sort of cultural activities. So there are many ramifications for infections, and the nervous system is a target. In fact, it's a really vulnerable target. And I'm going to talk to you about why the brain and spinal cord is a vulnerable target. So today, I want to just introduce you to a few aspects of the immune function in the brain, because it's very different from other immune structures or other organs in the body. A little bit about HIV, particularly HIV dementia, because that's a problem that we continue to encounter today, even though we have very good therapy for HIV. And I'm going to skip this neuropathy here. I was going to talk about peripheral nerve disease in HIV, and I'm going to talk about acute infections in the brain, because HIV is a chronic infection. I want to sort of contrast a chronic long-term infection of the brain versus a very acute infection, and that's actually herpes simplex, the thing that causes cold sores in the lips, can also cause a very rapid infection of the brain that can be lethal. So there are a few things. Inflammation is a feature of infections, and it was recognized almost over 2,000 years ago. And these are the sort of cardinal features: redness, pain, heat, and swelling. That's not necessarily the case. That applies to your skin. It applies less so in the brain. We do get swelling, and it can be lethal. Yes, people can get fever. Pain is not as big an issue in infection, and less redness in the brain occasionally. But in the brain, it's not always so clear. Is this really an effort to control the infection, or is it part of the disease process? And it isn't always so clear. But inflammation is very much part of the brain. You hear about inflammation in the body, but in the brain, it's become increasingly relevant in the last 20 years. We know that inflammation is part of the disease process in stroke and Alzheimer's disease, multiple sclerosis. And because inflammation is a changing dynamic process, it's lent itself to being something that we try to target from a therapeutic treatment angle. So a good example is multiple sclerosis. When someone has a relapse of MS, they get worse abruptly, they get maybe leg weakness. We treat it with anti-inflammatory drugs such as steroids, immunosuppressive steroids. So as I've alluded to here, it's a component of many diseases, HIV, dementia, multiple sclerosis, Alzheimer's disease, but also brain tumors as well. We can see inflammation in the central and the peripheral nervous system. The nervous system is different, right, from other organs. It's not as accessible. It's protected by the blood-brain barrier. It's got, and in the brain, it's surrounded by cerebrospinal fluid. So it's almost like a moat around the brain that protects it. There are no cells, or very few cells, in the cerebrospinal fluid. So the brain is very well protected, but nonetheless, it is accessible to infections and immune cells. And just to make the distinction, the inherent immune process in the nervous system, the brain, the spinal cord, and the peripheral nerves, is innate immunity. So there are resident immune cells that live in the brain, and I'll describe those resident immune cells. That's very different from the immune cells that protect you against infections like B cells that make antibodies. So when you get vaccinated against Hep B, we force your body to make antibodies against hepatitis B. That's part of the adaptive immune system. That's much more complex. It has a memory, not like a neuron in some ways. It's the B cell, it's the cellular substrate. But in the brain, it has its own resident innate immune system. And the innate immune system is much more primitive. You see an innate immune system in lower species. Goldfish have their own innate brain immune systems. Even flies have an immune system. Correct? Yeah. And there are different types of inflammation, and we'll get to that in a few minutes. But they're influenced by a lot of things. They're influenced by what is tickling them, the danger or stranger signal, because the immune system is there to respond to foreign infections or danger signals. And it's also very influenced by the genetic background. And that's something, I think if there's one concept you take away from this whole course is that genetics plays a huge role. Now we know the entire human genome. There are about 3 billion nucleotides in the human genome. We know all 30,000 genes, and they are different. Every one of you has a different component. And how you respond to infection is dictated in large part, or any kind of disease, by your own genetic background. So what are the key parts of inflammation? Well, it can be initiated from outside of the nervous system, but there can also be white cells that infiltrate the nervous system. It also can be if there's an infection directly in the nervous system, such as rabies, which infects the nervous system directly. You can get the brain gets activated and calls in white cells from the blood. The brain there's this interesting concept of it being immune privileged. And that has to do with the fact that it's protected. It doesn't have its own adaptive immune system. Don't worry about that too much. But what I do want you to take away from it, I don't know if you'll have lectures on Alzheimer's disease, but there's a very clear relationship between inflammation in the brain and the eventual development of neurodegeneration, neuronal loss. That's a relationship that's been really defined over the last decade or so. So activation of immune cells in the brain, like microglial cells, can lead to the death of neurons or damage to neurons. And that relationship we're ongoingly trying to define that relationship, but it's very clear that exists. That, if you go to the older textbooks, you won't see that, but it's pretty widely accepted now. Inflammation can have some good and bad properties. If you suppress it too much, there are going to be consequences, like you're going to get infections that you don't want in the brain. So patients with AIDS, their immune system's really depressed. Yes, they don't have much inflammation, but you know what? They are more susceptible to infections. What we're striving for when we take care of patients is to try and control the different types of inflammation. And that's something that many of us are actively working on now. Trying to be very specific about how we influence different components of inflammation. I would hope you guys know all this, but there are very specific cells in the brain, and trying to be inclusive, you could argue that any of these cells could be involved in inflammation. Neurons, terminally differentiated cells, are not usually thought of as cells that involve in inflammation, but they actually can direct inflammatory responses. Astrocytes, the most abundant cell in the brain, also they're very much involved in inflammation. These are the cells that we really, when you think about inflammation, you think about microglial cells and macrophages. Those are phagocytic cells. They're cells that originate in the bone marrow, and then they traffic to the brain. They are present in the yolk sac, and are present in the brain throughout development. But their brain is constantly being replenished with microglial cells or macrophages, and they come from the bone marrow. And they are really critical cells for cleaning up damage to the brain. Well, I'll show you. I've got a slide to describe. The peripheral nervous system is more or less the same. Slightly different series of components of cells. We have neurons, we have Schwann cells, and related satellite cells, macrophages. We don't have Schwann cells are almost like a chimera between a astrocyte and an oligodendrocyte. But I won't talk too much about that. So these are the major players. But I want to emphasize something that might not be emphasized in other lectures. Well, we talk about the blood-brain barriers protecting the nervous system, and I'm going to emphasize that. It is permeable, and all of us have blood cells that are trafficking through the nervous system all the time. They're there as in a surveillance capacity. They're looking for infections or damage to the brain that they can clean up. So macrophages are particularly, but also we have T cells that are involved in adaptive immunity. These are very sophisticated cells with memory for infections. They also traffic through the brain all the time. The dogma has been for many years that there's really not much trafficking into the brain, but indeed that's not true. We know today that there's constant trafficking, albeit at a low level. So yes, the brain is protected by all these important structures, but it's not absolute fortress. It is accessible to cells from the blood as well as infections, as I'll show you in a few minutes. So microglia are, I have to confess, a friend of my favorite cells. I know Kira really likes neurons. I don't know if you guys, you like microglia too. They're really dynamic cells. They're transported around, as I mentioned. They come in, they can be replenished from the blood supply. So in health, they're pretty quiet. You know, they have this ramified appearance. They're small cells. You know, this is actually a slight magnification. This is probably about 20 microns here, but they're smaller cells. They have processes. They're moving all the time. If this was a real-time video, you'd see them kind of sampling the environment all the time, moving their processes, and then they're looking for infections, they're looking to respond to neurotransmitters, they're looking to respond to growth factors. But in disease, where they get activated, such as an Alzheimer's disease or stroke, they get bigger. This actually, this is not the same mag, but they get bigger, and their cell bodies swell up, and they're called rod cells or mucoid cells. And those cells are really pumping out lots of inflammatory molecules, things like prostaglandins, cytokines, and chemokines. And those molecules talk to the nearby cells. And I'll show you some examples of that in a few minutes. So it's an incredibly dynamic process. And I know you've heard about neurotransmitters, but that's only one sort of subgroup of them. There are lots of other molecules that are being released that allow one cell to talk to another. Microglia differ depending on what part of the brain they're located. If they're in the cortex, they have a big ramified appearance. If they're in the deeper brain structures, they may have shorter processes, bigger cell bodies, and particularly if they're down here near the hypothalamus, they can be really big. And that may reflect the fact that the hypothalamus does not have, in there are many regions called the circumventricular organs that are relatively devoid of a blood-brain barrier. So these guys are constantly being activated, and that's why they've got that short, stumpy appearance, because that's the appearance, that's the picture or the sort of phenotype they take when they're activated. What do they do? Well, these are all, you don't need to worry about this. I'm just trying to make the point that they make lots of molecules that are potentially allow them to talk to neurons, to astrocytes. In many cases, they can be bad molecules, like tumor necrosis factor or excitatory amino acids, or a group of enzymes that we've worked on in my lab, matrix metalloproteinases. Cytokines, IL-1 is a notorious one. They're activated by immune molecules as well. Here's a T cell. could traffic into the nervous system and then activate the microglial cell. This is their list of functions. They clean up debris, they secrete molecules that call in other cells. That's called chemotaxis. They're also critical for infection. So they can take a piece of a bacteria or a virus and show it to the immune system in a certain way. It's called an antigen-presenting function. It can also be cytotoxic, toxic to neurons. There's been a lot of work at the University of Alberta in that area. They change, obviously, morphologically. They can replenish themselves within the nervous system, but also from without as well. So they proliferate, and they're able to mount very robust respiratory activity. So they produce lots of oxidative metabolism and its products. So they're very dynamic cells. They're not as stable as a neuron. Remember, a neuron, once you've got it, you're stuck with it for lifetime. It's stable, it's terminally differentiated. Microglia are very dynamic cells, and they're changing over time. Okay, microglia represent about 10% of the cells in the brain. The most abundant cell in the brain, and I know this is controversial, but the general, for your purposes, the dogma is that the astrocyte is the most common cell in the brain. About 89% of cells in the brain are astrocytes. And I'm showing you an oversimplified version. There are different types of astrocytes depending on where they're located. But they function very similarly to microglia. They are structurally important. They don't clean up debris as much. They do a little bit, but they're also susceptible to infection. They can make cytokines, inflammatory molecules. They're really key players in maintaining the homeostasis in the brain. Without astrocytes, you're in really big trouble. And there's some molecules that we can use to identify astrocytes in the brain, and if we have time, maybe we can talk about it. But these are very important players, and they're under-recognized. We live in an era, all due respect to Dr. Pearson, we live in an era of where neurons chauvinistic. We think about neurons most of the time, but let me tell you, this is the most abundant cell in the brain, right here. I think you guys think about neurons all the time, but those of us who are infection and immunity, we think about these astrocytes and microglia a lot more. So important group of cells. And then, of course, they talk to macrophages. They talk to oligodendrocytes as well. And, of course, astrocytes are critical for establishing the blood-brain barrier. Here's the blood-brain barrier. Here's a regular blood capillary. Just a few mitochondria. They don't have tight junctions. They just have tenostra. But in the brain, they're really solid structures. The endothelial cells have lots of microglial cells, or lots of mitochondria. They have what are called tight junctions, very unique proteins that prevent any sort of overt permeability. And then they're supported by glial cells around them, like astrocytes and microglia. You can't get an intact blood-brain barrier without those healthy glial cells like microglia and astrocytes around. So a good example of that is in a brain tumor. Do you guys have a lecture on brain tumors? You had it, so you know about how contrasts die on a CT scan or an MRI scan. It actually permeates. It can go right through the blood-brain barrier because the blood-brain barrier is not intact in a brain tumor. It doesn't have all these nice structural supports like astrocytes and microglia, ensuring that its integrity is preserved. So we can measure breakdown of the blood-brain barrier by looking at contrast scans, such as in a brain tumor. I think I'm going to move on. So I know this is a Neuro 210 course, but I want to sort of emphasize that you do need to think in a more integrated manner than perhaps you've been thinking. You do need to think a little bit about immunity. So I've mentioned this already, innate immunity. It's kind of the resident immunity in the nervous system. Endogenous factor, major endogenous cells in the microglia and the astrocytes. The exogenous cells, I didn't talk about this, but there's cells that can come in from the blood supply as well, that are more primitive cells. And then down here, adaptive immunity, and we talked about T cells. This is really all buzzwords for T cells, and various molecular mechanisms. Don't worry about it. I just want you to be aware of that. And if any of you take Neuro 410 down the road, you'll hear more about it. Key thing, I think I'll move on again because this is a little more than you guys probably need to know. I think I've addressed it. Okay. So this is more 410 stuff. This concept of antigen-presenting cells is, I think, pretty important for understanding disease mechanisms. There are different cells in the brain that are important for sort of showing the immune system infections or damage, and these are the main cells. These are the cells that sort of highlight those that damage or danger or stranger signals. They can be astrocytes, microglial cells, perivascular cells, which are other macrophages, and there are also other types of macrophages in the brain. There are macrophages in the choroid plexus. There are tons of macrophages in the covering of the brain called the pia and subarachnoid, and the dura. That is just loaded with macrophages as well. So macrophages are a big player in brain biology in general, and to some extent under-recognized. Okay, I mentioned all these. You don't need to remember these molecules, but there's a real spectrum of molecules that are active in the brain that participate in inflammation. I mentioned cytokines and chemokines. There are lots of enzymes like proteases and neuropeptides that are involved as well. This is a growing area. The list is by no means complete. There are lots more. But importantly, I think for those of you who read about systemic immunity, it's worth thinking about. If you take another course, say in cell biology or maybe one of the MMI courses, you're going to hear about a lot of the same molecules. They're at play in the nervous system as well. Perhaps not as well studied, but nonetheless they participate in brain biology in a big way, and often in disease. Okay, so that's a little bit of background about inflammation and brain biology and how it responds to infection. I'm now going to switch gears and talk about this passion of mine, which is infections of the nervous system. But you can't really understand infections without understanding a little bit about immunology, because the immunology is the way we defend ourselves against infection. So that's why I gave you that background. So infections in the nervous system are a little different from infections elsewhere in the body, because where it is in the nervous system really dictates it tells the clinician what it's going to be. So if someone comes in who's got say they've got a weakness in one arm, and we know, for example, that they say they've had a kidney transplant, so they're relatively immune suppressed. They come in with weakness in one arm, and maybe they've got a history of exposure to some kind of say cats, for example. Cats they harbor parasites called toxoplasmosis. So as a clinician, if I'm asked to see a patient who has had a transplant, they're a little bit immune suppressed, and they've got weakness in one arm, the right arm, I'm going to think, gee, this guy's immune suppressed and he's weak on the right side. He may have some kind of problem on the left side of his brain, perhaps an infection, and if he's immunosuppressed and he has cats, my first top of the list would be toxoplasmosis. And I would work down the list. I would think about other fungal infections. I would think about other viral infections. So where the region is, right away tells me what it possibly could be. That is not the case for other types of infections. The brain is unique in that sense. And if it's in the peripheral nervous system or the central nervous system, that further helps me delineate what kind of infection it is. We've alluded to this, the brain is kind of unique, it's so-called immune privilege. I think we've touched on the innate and adaptive immunity, how the infections might be dealt with. So, this is I like this quote, happy is he or she who gets to know the reasons for things. So, as a neurologist, I'm always really interested to figure out what's going on. And so neurology really lends itself there, because you've got to figure out, okay, based on where this lesion is, what could it be, and what could I do for this poor soul who's got an infection or some other terrible thing that's happened to them. So I like this quote because it kind of captures the spirit of things when we're trying to figure out what's going on with a patient. Okay, so brain infections are a little trickier to understand because they get access to the brain through different routes. It's not just like having a skin infection where chances are the bacteria or the virus got there through the blood supply. Not so straightforward in the brain. The virus or the bacteria can get access to the brain through accessing the peripheral nervous system and traveling within the brain, or it can get access to the brain through the blood-brain barrier, or it can get access to the brain through the choroid plexus, or it's just going to be free virus or bacteria that crosses the blood-brain barrier. There are a lot of different routes, and this is sort of highlighted. Importantly, viruses, most viruses, with the exception of rabies, they actually get into the blood, they travel through the blood, and then they get access to the brain through the blood-brain barrier. That's the general sort of Trojan horse approach to getting to an infection. But that's not the only way. A virus like rabies actually infects the peripheral nervous system and then travels up from the peripheral nervous system to the spinal cord, then to the brain stem and the thalamus. And so, it's done through retrograde transport as well. So there's several viruses that are unique to infecting neurons only. I mentioned rabies here. West Nile is another one that infects neurons. Herpes simplex, which I'm going to talk to you about in a few minutes, it infects neurons as well. And then mumps, chickenpox virus, varicella zoster, it infects neurons as well. Other viruses infect the myelin-producing cells, oligodendrocytes. Cytomegalovirus is a very common virus that we all have, all of us, that infects oligodendrocytes. Another virus called John Cumming virus or JC virus. HIV likes to go into these macrophages, and as well as CMV does as well. Astrocytes, HIV likes it, CMV, and the other human retrovirus called HTLV1 also affects astrocytes as well. And anybody, I don't know if anybody's traveled to India or Malaysia, you may know about Nipah virus, which is a virus that infects endothelial cells. You might have heard about that. It's a persistent epidemic that continues to recur, particularly in India. It originated in Malaysia and Singapore, through fruit bats. Now, one other thing, I'll just step aside from my role as an infectious person and think of, or neurovirologist, and just say our genome, as a result of sequencing the entire human genome, we now know that we have resident viruses in our genome all the time. So they're viruses that we've acquired over the last few million years, and they represent, extraordinarily, 8% of our genome. So these are viruses that are, for the most part, not infectious. So they're not transmissible from you to the gal sitting next door to you, but they do produce proteins, and in some instances they can actually cause disease. And they're, for the most part, called human endogenous retroviruses. So they're not unlike HIV, but they are not transmissible. But importantly, they can exert biological effects, so they can make proteins. And this is an area that we really don't understand, but let me tell you, there are lots of endogenous retroviruses expressed in the brain. And they've been in our genomes for as long as 30 million years, depending on the individual virus. We can stimulate them, turn them on, and they're implicated in a variety of different diseases. They probably modulate diseases. They're not disease-causing, but they increase inflammation. Maybe also protective, too. That's why we retain them in our genomes. Okay. So what about, I mentioned at the beginning emerging infections. I talked about Listeria, H1N1, SARS. So what about emerging neurologic infections? Well, some of you will have heard of the mad cow disease, and it was followed by what's called Creutzfeldt-Jakob disease in humans. You guys, I think, know the story of how mad cow disease was a disease in cattle in Britain. It occurred because they changed the way they prepared bone meal to feed to cattle, and as a result, those cows became infected with something called scrapie. It manifested as mad cow disease, though. It's a long and very interesting story. Well, there's a human variant of that as well, called Creutzfeldt-Jakob disease. And that was a disease that sort of sprung up in the early 1990s. It was mostly in young people, and there was a big scare in Britain about eating meat. It's more or less been controlled now, but that's an example of an emerging infection. We still see sporadic cases, but it's not very common anymore. I mentioned Nipah virus. It's an epidemic in Southeast Asia, West Nile, something that we've known about for almost 50 or 60 years, and then in 1999, it became evident in animals in the Bronx Zoo in New York, and obviously it spread probably by mosquitoes that were brought over in a ship or by airplane, and it got into the local population of mosquitoes in New York, spread to the zoo in New York, a bunch of animals died, and then it spread right across the continent, like wildfire in the subsequent years. So we had cases here in Alberta, we still have a case or two every year of West Nile virus infection. And it's we don't have a vaccine for it, but so it is sort of a recurrent emerging epidemic, and it's a bit concerning we don't have a vaccine for it. Enterovirus 77 is a virus that causes encephalitis, infection of the brain in children, mainly in Southeast Asia. Big problem, devastating outcomes. Most emerging infections, particularly viruses, are usually RNA viruses, because they can mutate really fast. A lot of these emerging infections occur because they adapt to a host fairly quickly and can produce disease. And RNA viruses lend themselves to that very, very easily. And we can talk about that. We have time after this. So I just want to talk, I'm going to talk a little more about HIV, because that's something that I think most of you know. There are about 35 million people infected globally. There's no cure for HIV. The drugs are okay. They're not great. Anybody saw Science or Nature a couple months ago? They sort of proposed that, I think it was Science, they said that just treating the disease will limit its infection. Yeah, that's true, but we don't have a cure. We don't have a vaccine. The vaccine for HIV has really been a big challenge. But if HIV is part of this family of retroviruses. So there's a very closely related monkey virus from which HIV is derived, simian immunodeficiency virus. If any of you have cats, there's a virus in cats called feline immunodeficiency virus. It's about 10-15% of urban cats have it, mainly males who get it from fighting. There's a virus in horses, a little scary, and it's in fact endemic in northern Alberta. It's called equine infectious anemia virus. And it's very closely related to HIV. It's in the wild horses up in the Peace River area. It's a problem. You might think, oh, well, it's off in the middle of nowhere. Well, guess what? It's transmitted by horseflies, and the racehorse industry is terrified of it, because if it gets into the racehorses, you basically have to euthanize the animals, because it's spread by an insect vector, so you can't treat it. So EIAV is economically important. And then there's some viruses in goats and sheep as well. Other retroviruses are HTLV1 is the other human retrovirus beside HIV. And then these are endogenous retroviruses. And then there are other viruses in mice and other species. These are called spumoviruses. There is also HIV too, and I'll just discuss that in a moment with you. So one thing I wanted to mention is you often will hear people who study diseases in populations who are called epidemiologists. They're often accused of over-projecting their disease. They'll say, well, there's going to be 100 million of this disease and 50 million, and they're often accused retrospectively, oh, you overinflated the numbers. But you know what? With HIV, they didn't expect it to be as bad as it was. In fact, they underestimated the numbers. And I'm showing you, this is an old slide, but it illustrated here, they predicted, this is a slide that I think I got in the mid-1990s, and they said that there would only be 30 million people who were infected by the year 2000. Well, in fact, it's closer to about 45 million people who were infected by 2000. It started to plateau a bit, but nonetheless, it remains a huge problem, particularly in sub-Saharan Africa. So be careful when you're reading about epidemiology, because often we're not very good at predicting what's going to happen, particularly in infectious diseases, because they can explode and be much harder than often people would predict. I'm going to skip that. That's the structure of the virus. So the origins of HIV are very interesting. HIV-1 comes from Central Africa. It's very clearly spread. It's not derived from a CIA conspiracy, as some people think. It's very clearly derived from the bushmeat industry. So people, hunters who are living in countries like Cameroon and Gabon and Central African Republic, hunt non-human primates for their living. And in the course of killing and cooking and eating them, they become infected with the simian immunodeficiency virus, which is, and in the course of making that transition from monkeys to humans, the virus becomes extremely pathogenic or disease-causing, just called virulent. And so this is a very good example of a xeno-infection from one species to another. In monkeys, interestingly, SIV does not cause disease. Those animals live normally. But as soon as the virus gets into humans, it immediately becomes extremely pathogenic or disease-causing. And this is the case for a lot of different infections. Measles is another example, and we have time to talk about that. But not all HIV are the same. There's HIV-1 in Central Africa here. HIV-2 is in West Africa, a little bit in Nigeria, Cote d'Ivoire, and it's much less pathogenic than HIV-1. It doesn't cause as much disease. So we've often thought maybe HIV-2 could be used as a vaccine for HIV-1, but there are really it hasn't worked out as well as we hoped. But HIV-2 remains a problem. We see it in Canada as well. But HIV-1 is the one that you hear about globally. It's the one in sub-Saharan Africa. It's the one in Canada, Europe, North America, India, and it's highly disease-causing. It's fatal unless treated. This is the main culprit. This is the main non-human primates, chimpanzees. People have been able to actually show that the transition from chimpanzees to humans occurred about 100 years ago. So it's a new infection in humans. It's been in chimps for thousands of years, just like the feline virus has been in cats for thousands of years. And as a result of that, monkeys have adapted to SIV and they don't get sick because they've been selected for, based on their genetic background, to tolerate the virus, or permissive to the virus. So today, this number has actually dropped. They're about 35 million, but there's still about 3 million new infections per year. This is a bit high. In Canada, we still see about 4,000 new infections a year. So right here in Alberta, we've got about 5,000 patients with HIV infection. Those patients are followed here at the University Hospital in the Northern Alberta Clinic or the Southern Alberta Clinic in Calgary. But the real concentrations of HIV are in Toronto, Vancouver, Montreal. And in part, that's related to the active drug activity. I'm sure you've all heard about the east side of Vancouver, where there was an epidemic of HIV about 3 or 4 years ago. What about the disease course of HIV? Well, it's a chronic infection. It's not like anthrax, which kills you, or which kills you very quickly. HIV is you become infected and you get an immune response in the first 4 to 8 weeks after infection. The virus goes up very high levels, maybe 1 million viral particles per milliliter of blood, then it drops, and it just kind of smolders along for many years, upwards of a decade in first world countries like Canada and the US. And then after about 10 years, the immune system is more or less had it. It's exhausted, and the virus just takes off and it's uncontrolled. And the marker for that, we measure blood cells in lymphocytes in the blood, and they diminish over time gradually, and then you reach a critical point where you really become susceptible to infections, and that's been termed AIDS. And there are various definitions of AIDS. There's an absolute white cell count in the blood as people use, but in Canada we use a definition of AIDS-defined names like HIV dementia or some particular infection, tuberculosis. But the good news is, when we introduce antiretroviral therapy, so-called HAART, highly active antiretroviral therapy, we can really control the virus. We can knock it down to undetectable levels. And that has been a revolution in care. I haven't seen too many revolutions. I've been in practice for nearly 20 years. I haven't seen too many revolutions in clinical care, but this is one of them. So it's gone from a situation where patients were literally dying all the time. I mean, it was a pretty ghastly in the mid-1990s. And today, patients are living almost like normal lifespans. It sounds good. There are a lot of side effects, there are a lot of adverse effects, but this has really been a revolution in care for patients, and it's saved the lives of hundreds of thousands of people. So, so we've learned a lot about AIDS in terms of other diseases as well, particularly in the nervous system. So in the nervous system, HIV affects the peripheral nervous system directly, and it can cause Guillain-Barré syndrome. It can cause neuropathy, distal sensory polyneuropathy, which is exquisitely painful, and we can actually cause muscle disease as well. You can find virus directly in the muscle. In the central nervous system, you can get meningitis because the virus infects those macrophages covering the brain. And so it can irritate the cortex of the brain and produce seizures. It also produces neurocognitive impairment, and there are varying degrees of that. So there's frank HIV-associated dementia down here, where patients are, and I'll describe that in more detail in a moment. There's often an antecedent condition called minor neurocognitive disorder. It's a bit like minor cognitive impairment that precedes Alzheimer's disease. I don't know. They have an Alzheimer's lecture. So there is a syndrome that precedes Alzheimer's disease where patients begin to lose their memory, they begin to lose their speech, and be able to perform different tasks. It's mild, so people can continue to operate reasonably well. That can progress into frank Alzheimer's disease. Similarly, with HIV dementia, it's often preceded by this minor neurocognitive disorder. And you can also see spinal cord disease or myelopathy, but we won't talk about that now. So the HIV dementia and the minor neurocognitive disorder have been collectively described the HIV-associated neurocognitive disorder. It's a horrible title, but that's what the literature is calling it. And it's different from Alzheimer's disease. In Alzheimer's disease, as I mentioned, you lose your memory, you lose your ability to speak, you lose your ability to carry out individual tasks over time. HIV, it's a little different because it doesn't affect the cortex as much. It tends to affect the subcortical structures. Remember I showed you a lot of those microglia where the subcortical structures? Well, that's where the virus likes to go. It likes to infect the microglia. So they're concentrated in the subcortical structures, so you get what's called a subcortical dementia. It's different from Alzheimer's, which is a cortical dementia. How does that present? Well, you can get memory loss, but it's a different kind of memory loss. You don't have as much declarative memory loss. It's other prospective memory loss. Maybe we have time afterwards. You get lots of other neuropsychiatric problems. You'll have apathy, sometimes frank mania. And then lastly, you'll get motor abnormalities as well. People will have tremor. They'll be unsteady on their feet. We call this ataxia. And the big thing, patients always tell me in the clinic, Dr. Power, I'm slowing down. I just don't think as quickly. I can't perform mental tasks. And I can't do anything as quickly as I want. And that is the cardinal feature. They just slow down. You know what? That's because it affects the subcortical structures. And does anybody have any guesses what are other subcortical structures that might cause movement disorders and dementia? Yeah, basal ganglia. Any particular disease? Yeah. You've heard of Huntington's disease, Parkinson's disease. Have they learned about vascular dementia, I guess?
No, later on.
Later on, so repeated strokes can produce that kind of picture as well. So it's the subcortical structures that are affected, not the cortex the way it is in Alzheimer's disease. That's all in the background of immunodeficiency as well. But you know what we're seeing today, unfortunately? We're seeing this constellation of features, these clinical features, even when people's immune systems are well controlled, which tells you maybe the drugs are not getting into the brain as well as they could be. And that's a topic for another day. How often does it happen? In untreated patients, it's anywhere from 10 to 20%. It's probably lower. It's probably about 5 to 8% of people who are treated. Even today, if you get dementia, your likelihood of survival is worse. It's usually, I mentioned, it's also called minor neurocognitive disorder or minor cognitive motor disorder. Same thing, this antecedent condition. We know that it's more common in children and also in older patients. The children, it's actually a really high rate. Of untreated children, it's about 50%. We call it encephalopathy. And they have developmental delay. They can have seizures. It's pretty ghastly, and it affects them for the rest of their lives. We also know their genetic susceptibility features. So we have a gene in this molecule called CCR5, which is the receptor for the virus. You have a mutation, you're protected. ApoE4, which is the epsilon 4 alleles, confers susceptibility to Alzheimer's disease. It also confers susceptibility to HIV dementia. And then we know about single nucleotide polymorphisms, where there's a single mutation, and it's actually in the promoter part of the gene, and two genes in particular, TNF, an inflammatory gene, cytokine, and a chemokine called MCP1. We know there's specific single nucleotide polymorphisms that confer susceptibility to dementia. So a lot of you will, in this room, will have these mutations, but because you don't have HIV, it's irrelevant to you. There are other synonymous terms for HIV dementia: AIDS dementia complex, HIV dementia, AIDS encephalopathy. The latter term is what we use for children. But good news is, in Canada, we have, I think there are 700 children infected in Canada. It's another incredible step forward. We can prevent infection mother to child today, because the drugs have become so good. We can treat mothers for several days before they give birth, and we can reduce the risk of transmission to less than 5%. Normally, it's around 30 to 40% transmission vertically from mother to child, but with the new antiretroviral drugs, we've been pretty successful. One of the good news stories is I think I've already touched on these issues, the behavioral changes, the cognitive and the motor function that define the HIV condition. I know you guys are going to get restless about quarterly too, right? That's right. That you guys have the capacity, so I'll keep on track here. HIV dementia varies widely in the severity. This is a measure of measure of severity of dementia. These are all patients that I took care of before we had antiretroviral therapy. They passed away, unfortunately. Some patients, the severity of their dementia did not change over the course of the year. Patient 19 was someone who was very stable in the year that I followed him between the time he was diagnosed to the time of death. And then in contrast, patient number 12, another fellow I thought I actually remember him distinctly. I saw him. He was, had a little bit of difficulty walking. His partner said this was in Baltimore, in Maryland, in the States. He was actually, I think he did a fair bit of cocaine, and his partner said, yeah, he wasn't quite right when I first saw him, but then in the course of 6 months, I saw him again several times, and this is before antiretroviral therapy. By the time I saw him again, he was basically bent down and almost immune. His dementia just had an extraordinary escalation of speed. There are probably multiple factors that dictate the progression of this disease. The type of virus you get, because the virus is extremely variable. It mutates all the time. That's why you can't get the vaccine. But it also is influenced by genetic background. So this fellow, unfortunately, number 12, was a guy who was almost living on the streets. He was poor. He was marginalized, bad nutrition, and he just did not tolerate it well. And number 19 here was a guy who was actually middle class. I think he died sad. I don't know how he got an HIV, but it was relatively stable and relatively mild. So a MSK score of 1 is someone who's almost living independently, whereas this 12 was bed down. So tremendous diversity in the presentation of the disease. It's dictated by the virus as well as the patient's genetic background. So what about today? Well, this is actually data we published a couple of years ago. HIV remains a problem in our clinics today. HIV-related neurologic disease remains a problem in our clinics today. This is work that a fellow postdoc in our lab published a couple of years ago. We looked at the risk of HAND, which is dementia and the antecedent minor neurocognitive disorder. Still represents a prevalence of around 15% of all patients. Pretty extraordinary. That's prevalence. So these are big. Remember, the prevalence rates are going up because people are living longer. Neuropathy, peripheral nerve diseases were also common as well. And the so-called opportunistic infections, the secondary infections because people are immune suppressed, were much lower than they were, say, a decade ago because of antiretroviral therapy. Unfortunately, for this group of patients, this is actually a cohort that we follow of about 500 or 600 patients. Many of these patients have multiple neurological disorders. So infection of the brain confers susceptibility to not just dementia, but sometimes seizures, sometimes spinal cord disease or muscle disorders. So it's, the virus obviously is not good to get because it makes you more susceptible to neurologic disease. But the most concerning thing that really bothered us the most is survival goes down. Even with antiretroviral therapy, if you have neurologic disease, your survival goes down a lot. And if you have dementia or the antecedent condition, again your survival drops. This is a so-called Kaplan-Meier curve. And you can see that people, if they have any kind of neurologic effects as a consequence of HIV, their survival goes down dramatically. So very concerning when we thought we had the epidemic sort of linked with antiretroviral therapy. I think I'll move on. Also, today in clinics, we worry not just about dealing with the medical issues, we also worry about people's quality of life. How do they cope with living with drugs? Because often they have to take multiple drugs. So quality of life is something we measure routinely. It's a very important issue. And the quality of life is really diminished if people have cognitive impairment. This is a self-reported scale, but it's been validated. And this is work actually a psychiatry resident did with me years ago. And he showed that if patients had neurocognitive impairment, dementia or the antecedent condition, their quality of life was really crappy compared to controls. If they had neuropathy or DSP, it was also reduced as well. So it's important to bear this in mind. This is a group of people with all HIV infected folk with and without neurologic disease. And it's pretty clear, as you might expect, the quality of life is diminished. The reason I raise this is something that if you go to the older textbooks, you won't hear about quality of life. But today, in clinics, we really worry about it. We want to make sure that patients, it's not just a case of dealing with medical issues, but dealing with the overall picture. This is a scale that actually I developed years ago when I was a postdoc that we used for HIV dementia. We still use it today. It's actually translated in Swahili and Japanese and German, so it's been a pretty good test. But what's important is it not only depends on memory recall or declarative memory, it also depends on psychomotor speed, the speed with which people can carry out different tasks, and also concentration. If I have time, maybe at the end I can tell you about what these different tasks are. The point is they're very different tasks than what you would get on a conventional bedside neuropsych test that for Alzheimer's or some other symptoms. Okay, this is actually just an example of a patient with HIV. Guy, actually, I still follow. He's a 38-year-old bisexual guy who we saw with a history of 3 months. He was known to be HIV positive. He had a 3 to 6 month history of increasing forgetfulness. As I mentioned earlier, he had slowed cognition, slowed motor activity, and he had difficulty concentrating. Couldn't read, couldn't even look at television and remember what he'd seen. In addition, his family said that he was more irritable, apathetic at times. That score, that test I showed you, the HIV dementia scale in the previous slide, if any of us in this room will get a score of 10 or better, so it's like we all get around 12, but this poor fellow had a score of about 6. And when I examined him, I found that he was unsteady on his feet. He was ataxic, so to speak. He had a tremor, not a resting tremor like Parkinson's disease, but what's called an intention tremor. So he reached out to something, he had a bad tremor. He did have a bad temper too. His reflexes were very brisk, so if I tapped his reflexes, his knees released or his toes shot up. And when he walked kind of sort of stooped and shuffled like Parkinson's disease. And that's a typical feature. So guess what? The basal ganglia is affected, right? Like Parkinson's disease. And he was profoundly immune suppressed. His CD4 count was 100 cells per microliter. Everybody in this room is probably about 1,000 cells per microliter of blood. So he was really immune suppressed. And he had about a million viral particles per mil of blood. So he was really, had very high viral load. So we treated him with antiretroviral therapy. And actually, he did incredibly well. That's the reason I'm showing you. He actually returned to work about a year later. It took a long time for the drugs to work, but he did well. And this is his MRI scan. And you can see here, I know that, unfortunately I don't have a control, but you can see first of all these dramatic white matter changes. That reflects disruption of the blood-brain barrier. But perhaps more importantly, you see that the brain is kind of atrophied. You see these huge ventricles. All of you in this room will have ventricles that are like tiny, like it's just a slit whereas this guy really has big wide ones. And in addition, his brain looks kind of like a pecan, sort of shriveled, or what we call atrophy. And that reflects a loss of brain cells as well. This, to some extent, is recoverable when we start antiretroviral therapy, but it's not complete. I have a patient who was very demented. And she has got antiretroviral therapy, but her brain really did not go back to baseline. She still has all this atrophy. And this is recent. This is in the last few months. And this is an old slide, but the antiretroviral therapy is evolved. We've made it more and more efficient and better, but we still can't completely reverse the process, unfortunately. What are the pathological features? What happens in the brain? Well, I mentioned these white matter changes. This is due to disruption of the blood-brain barrier. It's not due to demyelination. For many years, I worked on this when I was years ago. I thought it was due to loss of myelin, and it occurs in multiple sclerosis. It's not. It's due to altered blood-brain barrier permeability. Within the brain tissue, or parenchyma, you see lots of lymphocytes around the blood vessels, perivascular traffic. But this is the signature feature. This is a multinucleated cell. So this is actually a cluster of microglia and macrophages. Right here, you can see they've got multiple nuclei. It's kind of a ring, and that is loaded with virus. So the one thing you need to remember for this part of the lecture is that HIV infects the microglia. And that's a good, and the signature is the multinucleated giant cell. You don't really see it in many other diseases, this picture. And they're all macrophages. And if you look at the density of virus, because there's lots of virus there, it's mainly in these subcortical structures. So the caudate here, the globus pallidus, the cranium here, less so in the white matter, and really not very much in the cortex per se. It's all concentrated in these subcortical structures. So that fits with the clinical phenotype that it's a subcortical dimension, because that's where all the virus is. What happens to the neurons? Because the neurons are important, despite how I've ever dismissed by now. They definitely are damaged. So remember, neurons are critical for subservient movement and thought and concentration and emotions. So in someone who has AIDS, who's not demented, the cortex looks really good. You see these nice big pyramidal cells, lots of processes that look really healthy. You don't see any of this sort of death-associated protein P53. But in the patients with dementia, there are a lot fewer neurons, and they look dysmorphic. They look really unhealthy. And these are the big cortical pyramidal neurons. They also are often P53 positive, which is a sign of imminent apoptosis, or programmed cell death. You also, that's in the big pyramidal cells, but in the GABAergic cells, the calbindin positive cells, you see a relative loss as well here. That's often in the subcortical structures. You'll see a loss of the neurons, and just a few sort of scraggly looking neurons there. There are fewer of them, and they look really bad. So it's not surprising that people actually have difficulty with all those functions like memory, concentration, thinking, and movement. So you don't need to worry about this, except to know that a lot of us have worked on what part of the virus contributes to the disease. And my lab is interested. We've worked a lot on this protein VPR. We think it's a toxic protein. Dr. Kerr and I published a paper showing that actually this protein is also involved in killing peripheral neurons. Other proteins like the envelope protein, the TAC protein, they're secreted proteins. We think they're very toxic to the neurons as well. The virus, I won't go through its replication. It's much too complicated. But at the same time as the virus producing toxic factors, the virus also overstimulates the immune system. So remember I told you about cytokines, these inflammatory molecules. So the level of TNF-alpha, which is it's a cytokine that's really important in brain disease, including Alzheimer's, it's highly upregulated in patients with dementia. So it's not just the viral protein that causes the damage, it's also the exaggerated immune response. Too much of TNF-alpha actually kills neurons, and the specific receptor is worked out pretty well. You see TNF-alpha in non-demented brains and control brains, but it's much too high in patients with HIV dementia. I'm going to skip that. This is sort of a schematic model. So the virus infects macrophages. It produces inflammatory molecules and viral proteins, which causes apoptosis or programmed cell death in neurons. It also activates the nearby astrocytes, and they produce molecules that can damage the nervous system. But it's a dynamic balance, right? You need some of these molecules for homeostasis in the brain, but the virus perturbs them. The virus changes it, and it produces excess numbers of these molecules, such as cytokines, reactive oxygen species, which kill neurons and there's a positive disease phenotype. So I mentioned this. I mentioned antiretroviral therapy, HAART. Definitely, it works. We use it readily all the time. It's the cornerstone procedure. There's a lot of interest in using drugs that selectively protect the brain, such as there's a long non-competitive antagonist of the NMDA receptor called memantine. They know about the NMDA receptor. Glutamate receptor? You guys know about that? So memantine is a non-competitive antagonist. We use that in the treatment of HIV dementia. There's been a lot of interest in using drugs for conventional purposes. So there's a clinical trial now testing an antibiotic which has some putative neuroprotective effects. Minocycline, you guys may know it. It's a drug used for treatment of acne and various other bladder infections. It's now being tried as a neuroprotective agent. Inhibitors of TNF-alpha and platelet activating factors. A lot of interest in growth factors. Valproic was sort of thrown around as an anticonvulsant, probably not the case. SSRIs are also thought to be neuroprotective. But today in the clinic, I mean, when I see patients, I usually use memantine. And often if we have to use other drugs if patients are really agitated, antipsychotic drugs as well. I'm going to skip this in the interest of time and go straight to the acute infection. So I talked about HIV, which is a huge problem globally, but I want to talk about an acute infection. And don't worry, I'm only going to give you questions from what I talked about. So don't panic. I won't figure out the person in the other. So this is a rare disease, but it's a wicked one because it happens right here in Alberta. There are usually 2 to 5, usually about 10 to 15 people per year. Incredibly devastating disease. It's not, unlike HIV, it's not necessarily more common in immune-suppressed patients. We have very good diagnostic tools for it. I'll show you clinical examples, but it's fatal unless treated. It's the herpes simplex 1 virus, which can infect the central nervous system. We've now got pretty good drugs, so it's uniformly fatal unless treated, but with new drugs, or drugs such as acyclovir, we reduce the mortality to about 20%. So let me just say a little more about it. So herpes simplex is something we all know is a cold sore. It can be it's often on our lip. We get infected by it as children, but then it can be reactivated due to stress, studying for exams, getting a bad sunburn, and then it gets reactivated in the trigeminal ganglion, and it spreads to the brain. And I'll come back to that in a moment. Not unlike herpes, not like varicella zoster or chickenpox virus. Again, we get infected as children, but then as we age, we get older or we get stressed, we can get an episode of shingles. So you can get shingles on your face, you can get also a dermatoma strip of shingles as well, and that can be exquisitely painful. But in both cases, whether it's shingles or herpes on your lip, it's a reactivation of these herpes viruses. Herpes viruses are really stable. They've sort of been in humans and non-human primates for millions and millions of years. But they can get periodically reactivated and cause devastating disease. And what happens in the central nervous system, as I told you, herpes simplex can get activated in the gassarian or trigeminal ganglion, and then it spreads to the dura. This is one thought. This is not that well proven. It can spread to the dura, and then eventually it actually spreads to the temporal lobes in the brain. And this is actually a patient that we took care of. The patient
patient's wife, this is a man who was in his mid-50s. His wife reported, when we saw him emerge, that he was confused. He showed unusual lip smacking movements over the last 2 days, a little odd. So what was actually happening is he was having seizures. Had no history of a cold sore or anything, but his wife brought him to emerge because he was having these episodes of not being with it and lip smacking. So he came to emerge, and his only history in the past was high blood pressure. But when we examined him, we found that actually he was really disoriented, took person, place, and time. He had these atypical head turning movements where he was unresponsive, and that's called a complex partial seizure. And they usually arise in the temporal lobes. And during the assessment, he had his head turned, and then he actually that developed into a grand mal seizure where his arms and legs were shaking. He was clenching his teeth, and that lasted for about a minute or so. And then afterwards, he was really unresponsive. And so things were, once you get a seizure, like what these symptoms, things are really grim. So he was intubated right in the emergency room. We transferred him to the ICU. He had a CT scan, which I think I'll show you in a second, and he also had a lumbar puncture. Unfortunately, in this gentleman's case, he came too late. And we treated him with icyclovir, but he actually died, and this is the consequence. So you can see his temporal lobes particularly affected. They're really bloody. And the brain is hugely swollen. There's lots of so-called edema in the brain. And you can see that on MRI scan. If you recall that fellow with HIV dementia, you can see that this guy, in contrast, is very narrow ventricle. His brain is swollen. It's probably better seen on the CT scan here. And things did not go well at all. As I said, he actually didn't recover. And within the brain, you see inflammation, but it's different from what we saw in HIV. It's all these lymphocytes that are scattered through the brain tissue as well, around blood vessels, but also within the tissue as well. And you can also see viral particles very clearly. You can see it just on the histology. You don't need any fancy detection. You can actually see the viral particles. Unfortunately, that doesn't show up very well. These are actually viral particles within the neurons. Remember I told you HIV infects microglial cells? Well, herpes simplex travels through the trigeminal ganglion to the brain and infects neurons. So I don't know if you've had a chance to, have you heard about randomized controlled trials at all? So one of the things, the sort of gold standard for proving a therapy today, and I'm almost done with this, is to do what's called a randomized controlled trial, where you give one group of patients a drug, you give another, and on a random basis, and give another patients a different drug and no drug, so-called placebo. And one of the biggest advances in treating herpes simplex, or herpes simplex encephalitis, was the demonstration that acyclovir was actually beneficial. And this is a very old trial from Roger Whitley in the Journal of the American Medical Association in 1982. But he showed, and it's been much bigger studies, but he showed that when he compared Aracid, which is another antiviral, to acyclovir, he actually improved the overall survival. And it doesn't look great, but at this stage of the game, knowing that if you don't treat, the disease is fatal, this was a big step up in terms of treatment, from a 50% mortality rate to about an 80%, or survival rate, to about an 80% survival rate. That's a big step forward, and that's how we increasingly deal with drugs, it's an incremental change. So randomized controlled trials remain the gold standard for demonstrating drug efficiency, and you should be aware of that because you'll read about it in the literature. What else is on the horizon in terms of neurological infections? Well, you guys have probably heard about MRSA, the drug-resistant bacteria, a huge problem. Huge problem here in the hospitals, in the community. TB is another problem. We're having an epidemic of TB in our Inuit population in Manitoba right now. It's worsened by HIV. It's not an issue. HIV is not an issue in Manitoba, but TB is a huge problem. There are what are called iris syndromes, which are neuroimmune reconstitution syndrome, where patients are actually having kind of like an allergic reaction to an infection, which makes them worse. And I won't have time to talk about that. Globally, we're seeing a spread of infections. So dengue virus, which is a virus, anybody been to the Caribbean, like Dominican Republic or Cuba, dengue is a really big issue. But guess what? It causes encephalitis as well. and it's spreading to North America. It's now into the southern U.S. Likewise, malaria is also, it's on the spread. And the big concern, we don't have any drugs for dengue, and malaria, because we're running out of drugs because of drug resistance. So malaria causes a sort of like an encephalitis picture. I talked about influenza. This strain of influenza, H5N1, is very neurotropic. It can cause brain disease. The good news is, and I think I'm going to stop here and take questions, we have a lot of new technologies on the horizon. With the advent of large-scale DNA sequencing, we can identify viruses and bacteria, and we can also identify people who are more or less susceptible. We've got new microarray techniques that we can actually use in the field to detect infections so we can treat people earlier. So it's not all gloom and doom, but we're in an era of increasing infections and deaths. So I think I'm going to stop there, and I can take any questions. I'll hang around for a bit. Thanks for your attention.